the organizers, Alison, Anya, Andrew, and Anthony, for setting up this event and to keep it up regularly. So I think you make a really great contribution to the community here. Well, switching now to Chris. Well, I must admit I'm really lucky that he came to Graz in Austria 2.5 years ago to join my group as a PhD student. Before, he did his master studies at the University of Greifswald, and there he did his master thesis with Uwe Bornscheuer. And he also did his bachelor studies at the University of Greifswald. And actually, despite his young age, he has already done several internships. For instance, he had a 4-month internship in Japan at the University of Tokyo. And he also did a about 5 months internship in Canada at the University of British Columbia. In his PhD, or during his PhD, he's dealing with ether formation and ether cleavage, especially methyl phenyl ethers. But he's not using there for the cleavage, as you will see, and that's what he's talking today about, a different enzyme than normally used, so not an oxidative enzyme. So with that, I would like to hand over to Chris to give his talk.
Thank you, Wolfgang, for this great introduction. So today, welcome, ladies and gentlemen. Today, I would like to talk about my PhD project, and I'm working with cobalamin-dependent methyltransferases, which can demethylate a methyl ether group without oxygen. So first of all, why do we need to cleave the methyl group from this methyl phenyl ethers? And for the CO bond, for example, we have a very high energy barrier, like 358 kilojoule per mole, to do this cleavage. And normally for the chemical demethylation, we have very harsh reaction conditions, which are also rather environmentally unfriendly. For example, there we have to use strong acids or bases. We can also apply metal catalysts, which are from time to time expensive, as well as nucleophilic thiolates, which are quite smelly substrates, and also oxidative and reducing reagents. So where can we find all those demethylated products actually? We can find these products anywhere in our life. Basically, we can find it in our bathroom. We can also find it in our kitchen as a flavoring compound, as well as we can find it in our garden. For example, if we want to get rid of some insects or plants that we don't want to have. As well as we have it in the red dyeing compounds, such as alizarin. And also, once in a while, if we have to drink a glass of red wine, we can also find it there. As well as catechol, which is a main building block for new pharmaceuticals and drugs. In my presentation, we will also take a look onto these two natural products. The racemic yatin, which is a rather large compound with 3 methoxy groups, which is a precursor of podophylotoxin, which can be used to treat warts, as well as papaverine, which contains 4 different kinds of methoxy groups, which can be used to relax our muscles, and also which can also inhibit the human prostate cell cancer growth. So what can nature give us to challenge these demethylation specifically? Because the chemical side is also not really regioselective, it's not environmental friendly. So therefore we have enzymes. Oxidation oxidative enzymes, for example, for the demethylation can be used. We have D and monooxygenases, as well as fungal peroxygenases. If we take a closer look to this P450 monooxygenase catalytic cycle, we easily see that oxygen and hydrogen peroxides are involved. Hydrogen peroxide is in high concentration, really difficult for enzymes because they can easily be degraded, as well as oxygen is a problem because oxygen, if we take at this look at these catechol building block, this can easily be oxidized to the autoquinone intermediate, which will further lead on to various products such as known as this black precipitation in your epi. And it is also stated in literature that if we have this 1,3 benzene diol, in this paper they found out that we have several dimerized products for this oxidation process. So to analyze everything, it's a nightmare. So therefore, our research kicks in. We have cobalamin-dependent methyltransferases from anaerobic bacteria, which don't need any oxygen at all. So with this, we have not the oxygen problem. And in nature, how does this cobalamin-dependent transferases can do the reaction? So there's a complex reaction mechanism. So first of all, we have two methyltransferases, the MT1 and MT2, as well as the carrier protein, which is loaded with cobalamin or methylcobalamin, which can be shown here in this picture, which has a really nice red color. And the carrier protein acts as a methyl shuttle. In the first part, the substrate can be accepted by the methyltransferase with the help of the carrier protein to produce the hydroxylated products. On the other hand, we also have methylation process occurring, taking the tetrahydrofolate as a methyl acceptor, leading up to the methylated tetrahydrofolate with the methyltransferase 2. If oxygen is involved in the whole system, the active cobalamin 1 can easily be oxidized to the inactive state cobalamin 2, but therefore nature also has a solution. Therefore, we have another enzyme, which is the activating enzyme, which takes ATP and 1 electron to reduce the cobalamin 2 back to its active state cobalamin 1. So far, so good. If we have this kind of complex system, it's very difficult to set up a biotransformation. That's why we had the idea to just simplify the whole methyl transfer complex down to one enzyme, which is the methyltransferase or the VDMB. Another name for VDMB is the veratrol O-demethylase because veratrol is used as a natural substrate. As well as we have the carrier protein DH4611. And as you can see, those two proteins derive from different kind of anaerobic bacteria. The VDMB can accept a bunch of substrates, such as we have always these two methoxy groups in a moiety in close proximity or in 1,3 position to each other. Then we will, of course, lead to the hydroxylated products. On the other hand, the VDMB can also do the methylation of the co-substrate. The good thing about it is that I am working in the glove box, so we have not any oxygen involved in these processes. So we have not any oxidation problems at all. In our reaction, first we started with veratrol, leading to these products guaiacol, as well as different kinds of methyl acceptors, such as the dihydroxybenzaldehyde, as well as orcinol here. And as you can see that we have an excess of these two methyl acceptors compared to the methyl donor. This is why, so we need an excess of the methyl acceptor to push the reaction equilibrium towards the demethylation. So, and my researcher also investigated a lot of different kind of substrate scopes. First of all, veratrole is accepted with 55% conversion, leading to guaiacol mainly. So there we also have like a mono selective demethylation going on, as well as very tiny bits of double demethylation going on. Then if we just incorporate different substituents, such as a methyl group here, then we will have the highest conversion regarded so far with 77%, leading to this regioisomer preferably over the other one, as well as a double demethylation. If we increase the temperature to 35 degrees Celsius, we can see that the conversion is decreasing to 62% instead of 77%. But we also see that the double demethylation is even getting higher. So taking a closer look onto that, we did a time experiment for 20 hours. And in the first beginning of these 2 hours, for example, the green monodemethylated product is preferred by the enzyme over the blue products, as well as after 6 hours, if these monodemethylated products are accumulating, we get to the double demethylation of these products. Then for entry number 3 with the aldehyde group, there we find a very high regioselectivity for the vanillin, the flavoring compounds, as well as entry number 4, we see remarkably regioselectivity for this product and not any other product at all. And then we also figured out what if we just test like 2 methoxy groups in 1,3 position to each other. And interestingly also here with entry number 1 with the 3 methoxy groups as a substrate, this could also be accepted. We cannot observe any triple demethylation, but we also have a rather high double demethylation going on into this process. If we, for example, take this monodemethylated product in the next entry, we see that we have a regioselective monodemethylation going on. So at a certain time of point, the demethylation stops. For entry number 3, we have this 1,3 methoxy position to each other, and we find also a very high regioselectivity of the enzyme. And if we incorporate the methyl group towards the substrate, we could also see that one regioisomer is preferred over the other one. Now, also with the natural products, with the yatine, that's a rather promising approach because we want to have larger molecules. We also want to apply them for pharmaceutical use, and therefore we use the racemic yatine, which contains these 3 methoxy groups. And the racemic yatine is a precursor of this podophylltoxin, which is a treatment against warts, which can be obtained by this plant, the Podophyllum peltatum. And the veratrole O-demethylase easily demethylates here in the middle position to this racemic 4-D-methyl yatine. For the papaverine, the next larger molecule, this is a secondary metabolite of the opium plant from Papaver somniferum. The papaverine can be demethylated here regioselectivity and with the 6-desmethyl papaverine, we could mostly obtain so a 23% conversion. And also we can see that a double demethylation is going on to this 4,6-D-desmethyl papaverine, where we only got a very poor conversion with 2%. Interestingly, these 2 products are valuable biomarkers to improve the detection time of the drug heroin. We also did a semi-preparative scale of the substrates, and we could isolate the substrates and the products and could verify these products over 2D NMR experiments. So to summarize everything with the oxygen-free methylphenyl ether cleavage, the good advantage about is that no oxidation problems are involved, so we don't have any formation of various products, which is a nightmare to analyze. Then we have an easy facilitated methyl transfer concept. So we reduced it just down to one enzyme, the methyltransferase, and one protein, the carrier protein, which acts as a methyl shuttle. Then the veratrol O-demethylase is selective, depending on where the substituent is located on the aromatic ring. As well as we have a selective monodemethylation going on. Of course, we also have double demethylation, but this enzyme prefers and stops most of the time at the monodemethylation reaction. Also, natural products were accepted, like papaverine and yatein, which are very valuable for pharmaceutical industry. And yeah, with this, I would like to acknowledge the ALK crew where I'm working with. Special thanks goes to Wolfgang for being such a great supervisor and giving me this very challenging but really interesting topic. Thanks Michael and Matthias for helping me out with the 2D NMR experiments, as well as my great colleague Simone for also giving me advice on chemical issues, as well as Johanna that also contributed a bit, and Judith and Nina, who were my former working colleagues. With this, I would like to thank the audience and are open to answer any question.
Hey, great. Great talk, Christopher. We definitely have time for a few questions.
Yeah, thank you so much, Christopher. This is actually a fascinating enzyme. Can you comment a little bit on the substrate scope, as well as on the kind of diversity of those enzymes in nature? Can you use some aliphatic substrates or sugars instead of the aromatic polyphenols?
Actually, this methyltransferase, the veratrol O-demethylase, is only selective for aromatic substrates, so no aliphatic substrates are accepted. And also the two methoxy groups are very important to set up the demethylation, as well as for the methyl acceptors. We also have like the kind of flexibility where these two hydroxy groups are located. So there's a little bit of playground, as well as also with larger molecules. So we also got the crystal structure of the veratrol O-demethylase, and it has a very large binding pocket also to accept larger challenges in that case. Yeah.
And with regards to this one electron activation of the inactive enzyme, so is that also enzyme catalyzed? Is the enzyme kind of part of a gene cluster, or how did you elucidate that?
Sorry, Mr. Electron, can you repeat it?
So you showed there that that if the enzyme goes into the inactive coupled 2 state, yeah, then you need an electron from an ATP-dependent enzyme, I presume. So is this enzyme part of the same gene cluster? How did you elucidate that?
So that's also a good question. That's also in the same cluster of the Acetobacterium dehalogenans, which is the bacteria. And this contains like an activating enzyme under depletion of 1 ATP and 1 electron. And this step is crucial if only oxygen is involved in nature. Then the inactive state of this cobalamin cofactor can easily be reduced to the active state cobalamin 1. So in our case, with this facilitated a concept, we don't have that anymore because I'm working in a glove box without any oxygen involved, so which is a great advantage, because otherwise we have to monitor so many proteins in these reactions.
Sure. And we have one question from Peter Wanyama online about what can you do to improve the yields, and can you comment a little bit on the regioselectivity that you observed, that sometimes it was very selective, sometimes it wasn't?
Yeah, so basically with the regioselectivity, it depends on the aromatic ring. So for example, we have benzene, and either we have at least 2 methoxy groups, which are required for the demethylation. And then, for example, if we have 3 or 4 methoxy groups, that can also be demethylated. But of course, also if we have other natural products which are very, very polar, then this can also inhibit the demethylation process. So it also depends how the orientation of this benzene ring is binding to the cavity. Yeah.
Great.
Thank you.
So if there aren't any further questions, we can thank Christopher again. And I guess we'll move on to our final talk today. So